let us get started. And what I wanted to start with today is recapping, or I guess giving you time to ask questions about glycolysis, redox reactions. I'm not going to go through every part of this, but I do want to make sure that we're able to, for example, identify the different types of reactions that we see throughout aerobic respiration, anaerobic respiration, and fermentation. All right. And again, I guess I should probably reiterate, why are we taking the time to go through this? And in part, one, this is how every single microbial cell produces energy. Two, this becomes the basis for growth requirements, growth control. And then also, this becomes the basis for metabolic tests that we use to identify microorganisms as well. Some of that diagnostic testing that we've begun to do in lab. So I'm going to pause. What questions do you have? Annie.
Redox reactions, the very part.
Yeah, right at the end.
I feel like redox in like Gen Chem is different than this.
It kind of is.
Okay.
Yeah, let's take a look at a couple of these and talk about how do redox reactions occur here. So you may remember that on Wednesday, one of the things that we talked about was there's an oxidizing reagent, there's a reducing reagent, and there's an oxidized product and a reduced product. In other words, if we kind of draw this out, the way that the book kind of models it, this is definitely not black. So if this is a molecule that has free electrons, and then we have a second molecule here, the redox reaction would involve the movement of these electrons from one substrate to the other, or one reactant to the other, resulting in two new products. The one that has lost its electrons, is that oxidation or reduction? That's oxidation. So this we would refer to then as the oxidized product. The one that's gained electrons is obviously then reduced. These are always paired reactions, at least when we look at it full like this. What else moves with those electrons? What else typically moves in a biological redox reaction?
Hydrogen ions?
Yeah, hydrogen ions are going to be moving as well. In some way, shape, or form. They may bond here, or they may just be lost, and we see that being the case in the electron transport chain. And if we look at this half over here then, the one that is being oxidized is giving away electrons. In other words, it's reducing the other reactant. So we can call this the reducing agent or reducing reagent. And the one that is ultimately going to be gaining the electrons or taking them away is the oxidizing agent. So that's just the general overview. Go ahead, Annie.
So follow up, if you're seeing hydrogen ions move, do they always move with the electrons?
You can assume so,
Okay.
yes.
Okay.
Yes. So taking the first example up here, then it's NAD plus, or nicotinamide adenine diphosphate, going to NADH. The H here represents the hydrogen. Is this as drawn an oxidation? We would describe this reaction as an oxidation or a reduction. This is a reduction, yeah, exactly. So the NAD plus is gaining electrons. It's actually gaining 2 electrons and 1 hydrogen, typically in these reactions. What's not shown is there's an extra hydrogen kind of free floating at this point. That would be a reduction reaction. FAD to FADH2, same thing, another reduction. And remember, these two molecules, or these two pairs, I suppose, NAD and NADH, and FAD and FADH2, these are coenzymes, okay? They are carrying high energy electrons in their reduced form, and where do those electrons eventually go? Where do these ubiquinol? This one occurs right in the electron transport chain. I'm not asking you to know the name ubiquinone and ubiquinol. Actually, you don't even need to know what it is to be able to answer this question. Focus on the FADH2. The reason why I'm giving these 4 particular examples, these are ones we see commonly within metabolism. The major molecules that are involved in redox reactions in prokaryotes: NADH, FADH2. Some photosynthetic ones are going to use NADPH, but that's not something we're going to cover. And then there's one other place that we see redox reactions, and that's the electron transport chain. So we'll spend a little bit of time looking at that later today. Annie, does that help to answer the questions? Okay. Other ones that folks have.
So sorry, just to clarify.
No, that's
with
fine.
Number 3.
Yeah.
It's the oxidizing agent then is is that 80 plus? Or is
Yes,
that, okay.
yes, sorry, I'm sitting there trying to figure it out as you're saying. Yes. So the oxidizing agent is the one that is always paired with, we can say that it's paired with the reduced product. Allie, did you have a question?
Yeah. Can you go over the overview question, number 2?
Oh, sure. Okay, so how many substrate level phosphorylations occur in glycolysis? That's a great place to start, actually, because we're going to compare that with oxidative phosphorylation today. You may recall one of the things that we talked about on Wednesday was that substrate level phosphorylation really refers to the movement of a phosphate from one reactant to another to generate 2 new products. So if we can just relabel these terms, actually, and the general equation looks exactly the same. So in a substrate level phosphorylation, we've got some sort of phosphate group, again, not black, being moved from one of the reactants to the other to produce 2 new products. Okay? And oftentimes, at least when we're looking at this in the context of glycolysis or even Krebs cycle, the one that is, at least on one of these sides, we've either got ATP as the reactant that contains the phosphate, or we may have ATP as the product that gains the phosphate. Okay, so let's take a look at a couple of examples here. What kind of enzymes are involved in this phosphate transfer? Kinase is the major one. So let's go back, let's go to the top of this here. Here is our process of glycolysis, and in glycolysis we've got a couple of points at which we see substrate level phosphorylation. Typically when we're talking about substrate level phosphorylation, we're talking about which are the steps that help us to gain ATP. But we can look at it both ways. Okay? So which reactions or which steps here contain or involve an ATP either being destroyed or made? And just, you can yell out the numbers as they appear on the sheet if you want to. Sally, can you identify one?
Step 1.
Exactly. So we've got a phosphate on it, we've got ATP. It's going to donate one of its phosphates to glucose to form this molecule here, which is known as glucose 6-phosphate. That's absolutely a substrate level phosphorylation. Where are there others? Step 3, fructose 6-phosphate to fructose 1,6-bisphosphate. So now this one has 2 of the phosphate groups. Which of the substrate level phosphorylations here give us ATP in the end? Somebody said this one right here. I don't remember the names of these anymore at this point. But we're getting 2 ATP at this point because we have this molecule here splits into 2, and everything from here on out is occurring in duplicate. So we're yielding 2 ATP from 2 reactions there, and again over here. Okay? And you can bet that the enzyme involved in both of these steps are more kinases. This one is known as phosphoenol pyruvate, or no, that's not the name. Something kinase, pyruvate kinase or something like that. So substrate level phosphorylations, creation or destruction of ATP, the phosphate's always being moved from one molecule to another. Now I'm going to draw a quick contrast here because I want to, since we're on this topic, I want to compare this to oxidative phosphorylation. In oxidative phosphorylation, what we find is this following reaction. The PI here just refers to inorganic phosphate. You can think of this as free-floating. The difference between these two reactions is phosphates here are always bound to one of the two substrates, and then stay in the system, are bound to one of the two products. In this example here, this phosphate's not bound to anything. It's free-floating. So that's one of the key distinctions between these two mechanisms. There's a transfer of a phosphate versus the addition of a phosphate from the air. Do you want to go through more of those questions, Allie?
No, I think that's good.
Okay, okay. How about redox reactions? Where are there redox reactions occurring in this process? One. Why? What are the characteristics of a redox reaction?
Something being added.
What's being added? Okay, electrons. And what did we say moves typically? Electrons and?
Hydrogen ions,
But what's being added in step 1?
the phosphate.
Does that fit with? No. Okay, good. Good. Where do we see a redox reaction?
6.
Yeah, somewhere right here, right? It's kind of hidden, but we've got, so what this is showing is, again, phosphofructose, or once phosphofructose bis, blah, blah. Fructose with 2 phosphates. Gets split into 2 different 3-carbon molecules. One of them gets converted into the other. Same molecular formula, different molecular structure. What kind of enzyme is driving this reaction? Isomerase, same molecular formula, different structure. And look what we have. NAD plus is being converted into NADH. Is this a reduction or an oxidation?
Reduction.
That's a reduction. In other words, this 3-carbon molecule here is being oxidized. Okay? And we get 2 NADH from this process. So there's one of the redox reactions that we see throughout this. And that's actually the only one that happens in glycolysis. Additional questions? Other steps that folks want to kind of review right now? Okay, let's switch it up a little bit. Just set that aside for right now. And again, I'm not asking you to memorize this process of glycolysis. I'm not even going to ask you to memorize the Krebs cycle. But what I want you to do with this figure here is you got to tell me, where are we getting, what kind of reactions are happening here? Are there substrate level phosphorylations, or are there kinases involved? Where are there dehydrogenases? Is there potentially, there is, but I don't know if we can tell it exactly from this one. Is there an isomerase being involved at some point in this reaction? So I'm going to give you a few minutes, we'll say like 3 minutes, and try to identify as much as you can in this reaction. Okay? What's the theme of this? What's the theme of, if you could summarize, what kind of reaction is most prominent here? What kind of reaction? Redox reactions. And that's the theme of citric acid cycle, Krebs cycle, tricarboxylic acid cycle, whatever you want to call it. I don't care. But the main goal is redox. In this particular set of steps here, what we have, and this doesn't do a great job of showing it, we end glycolysis with pyruvate. Some folks will call this a transition step. Pyruvate loses CO2. That's the fully oxidized form of carbon, is carbon dioxide. That oxidation couples with the reduction of NAD to NADH. And then the acetyl-CoA that's produced goes right into this cycle here to produce first a 6-carbon, 6-carbon sugar, and then we've got a 5 and then a 4-carbon sugar towards the end here. And that's what those 3 different colors represents. Where are our redox reactions? What are we looking for? Hydrogen moving is a great answer. So where are those steps?
They're the isocitrate.
Isocitrate to alpha-ketoglutarate, that's one. Alpha-ketoglutarate to succinyl-CoA, there's another. And look, each of these is paired with a CO2 loss as well. That's that fully oxidized form. Succinate to fumarate, this occurs right in the bacterial cell membrane. We'll come back to that in a bit. And then malate to oxaloacetate. Remember, how many times does this cycle occur per molecule of glucose? Should occur twice, because we get 2 molecules of pyruvate out of glycolysis. Now, we always view this as a cycle. This is kind of the classical way of looking at this. One of the things to point out here about this cycle, though, is that there's a lot that feeds into it, and there's a lot that feeds out of it as well. So, for example, if a cell is in need of making amino acids, alpha-ketoglutarate is kind of a crux point for that. So if a cell, instead of needing to make ATP, is requiring the production of amino acids, alpha-ketoglutarate becomes a precursor to some of the amino acids. And so from this breakdown or catabolic process, we start to view the buildup of building blocks for macromolecules. Or if there's a carbon source other than glucose, if an organism is eating a lot of amino acids, feeds in right to this step and then goes through the cycle. So the point is that this is an enclosed system. Okay? And it's not as if you were looking at a cell and this thing actually does happen in a circle. Those are just how we view them, right? Rather, it's much more complex than what we're viewing here. The only isomerase step in here, not that this isn't really that important, because again, we were saying the NADH is the big one. This right here, citrate to isocitrate, it requires the same enzyme, aconitase. This is an isomerization reaction. Okay? That's something maybe you can wow your parents with over the weekend. Hey, or kids. All right. Where does this go? What happens at this point? We've got all this reduced NADH and FADH and a couple of reduced FADH2. What happens to it? What's that? I didn't hear. I honestly did not hear.
Does it recycle back in?
Recycle back into where?
Glycolysis.
So you're saying, so in order for it to recycle, what has to happen? The NADH has to go back to what?
NAD+.
NAD+, absolutely. But how does it get recycled back to that form?
Electron transport chain.
That's where we've got it. So the electron transport chain is going to provide that ability to oxidize NAD and NADH, or NADH to NAD and FADH2 to FAD. So let's take a look at that. Bacteria don't have mitochondria. No membrane-bound organelles. Where does this take place in a bacterial cell? Cytoplasm is where glycolysis would take place in the Krebs cycle. Where does this take place? This event, the electron transport chain.
In a membrane?
Yeah, cell membrane. Okay. So what I like to do here is draw a comparison between the mitochondria, or what you know about the mitochondria, with what we know about a bacterial cell structure. And let's think about a gram-negative here. So what does the cell envelope of a gram-negative look like? They've got an inner membrane. Then what? Thin or thick peptidoglycan? Thin layer peptidoglycan. And then what's outside of that on a gram-negative?
Outer membrane?
There's an outer membrane. What's the structure of a mitochondria? There's a lot of correlation here, and actually there's strong evidence to support that mitochondria are derived from gram-negative bacteria that gained some sort of endosymbiotic relationship with eukaryotes. We can look at mitochondrial DNA, and there are genes in here that are very similarly related to genes that we find in some bacterial species. So the events that take place in the mitochondria take place in analogous locations in gram-negative and gram-positive bacteria. Primarily, that electron transport chain is located on that inner membrane. So as NADH comes in, first step of the electron chain is that NADH will give off its electrons, give off hydrogen. It's undergoing an oxidation. That means that this complex right here, what is sometimes known as complex 1, the other name, NADH dehydrogenase, it's gaining electrons. It's being reduced. And those electrons, in this particular electron transport chain, what we find is that these electrons will either go to additional coenzymes. So FMN, for example, is one of those coenzymes. The other location we might find those electrons going is actually to cofactors, in particular iron, or Fe3+. And as iron picks up those electrons, it's going to go to a reduced form, which is Fe2+. The details of that, of like 3+ and 2+, not that important. But knowing that iron is an integral part of this is important. Iron is what we oftentimes consider a limiting reagent. It's required for bacteria to live and grow, and oftentimes they're fighting with other bacteria or even our host cells for that particular molecule. So where do the electrons go after they're in that first complex? What's next? Carson, you moved your pencil. What's that mean? I can see everything. I also can hear everything too, just so you know.
Just move down, just keep moving down
Moving down where?
to the different, like, proteins and the different, like,
Or complexes. That would be, that's good. That's good. Okay, so they're moving from one to the next, right? As this one right here gives off its electrons, this is being what? It's losing electrons. So is it being oxidized or reduced? This one's being oxidized. This one is being what? Reduced in the process. And then it hands off electrons to the next complex. Another redox reaction. Hands-off electrons again, another redox reaction. So on and so forth. The electron transport chain is full of these redox reactions. As those electrons are moving, they're losing energy. Okay? And as they're losing energy, how is that energy being used? What is that energy driving? Not just yet. Not ATP just yet. There's something that has to happen before the ATP gets produced. What do you see here is moving from inside the cell to outside of the cell? Yeah, that's the purpose of those electrons. Those high energy electrons, and I know a couple of people actually put this into their response. This is active transport. This is using energy to pump electrons across, to pump hydrogen ions across a membrane from low concentration to high concentration. pH of the intermembrane space in mitochondria, about 4.5. Not that low in this periplasm space in bacterial cells, but the pH here is south of neutral. It's lower than a neutral pH. Inside, it's somewhere maybe just a tad above neutral. And that helps to support the observation too, the pumping of those hydrogen ions against their concentration gradient. Okay. Now how do we get the ATP? What do those hydrogen ions do? Go through ATP synthase. Active or passive transport at this point? Going from high concentration to low concentration. This is passive transport. And that passive transport, that chemiosmosis that we observe, that's what's driving the oxidative phosphorylation, or this reaction right here. ADP plus inorganic phosphate forms ATP. The electrons, they don't stay in the chain. Where do they go at the end of this process? They do go on oxygen, if we're talking about aerobic respiration. Okay? So we'll come back to that in a minute. But maybe a more general way to phrase this would be, they go to a final electron acceptor. Oftentimes we think about this in terms of oxygen, because honestly, out of all the final electron acceptors in this process, oxygen is by far the best. It allows for the most amount of energy of those electrons to be released, and ultimately leads to the production of the majority of the more ATP than any other form of respiration. Questions on this process? I'm not concerned with you necessarily knowing how much NADH gets produced or how much FADH2 gets produced. I'm not really even concerned with you knowing the exact amount of ATP that gets produced. Those aren't really important, trivial facts. What's more important is understanding how does this process occur and why does this process occur. So the electrons, for example, they're losing energy and they're continuing to be pulled from one to the next because of a property that we've already talked about. If a molecule or an atom has a pull for electrons, what's that property known as? Go back to week 1, if you can think about it. It's related to polarity. Yeah, electronegativity. As those electrons are moving from one complex to the next, each of these molecules that accepts the electrons, it's more electronegative than the previous one. So it's not as if those electrons are just freely moving. They're actually being pulled from one to the next. Oxygen in this set here is the strongest, is the most electronegative. And that allows for the greatest amount of release from those electrons throughout this process. Okay. We'll compare this in a minute with different forms of respiration. Questions on the process? Okay. Let's take a look at I'm going to skip this next slide. This really is not important, but I put this up here because I love the image. So there are different forms of respiration. Aerobic respiration obviously refers to oxygen being used as a final electron acceptor. The alternative, anaerobic respiration. One of the things that oftentimes is tricky is when we think of respiration, we think of what we do. Breathe in, breathe out. And we think, yep, oxygen is being used. Respiration doesn't refer particularly to oxygen being used as a final electron acceptor, but respiration refers to the process of Krebs cycle and electron transport chain and oxidative phosphorylation. So if a cell is generating ATP through the electron transport chain, we can define it as performing respiration. What's different though is what is that final electron acceptor? And this is where we see the dichotomy between aerobic and anaerobic. So aerobic, oxygen used. Anaerobic, something else. There are three very common final electron acceptors, and we find this in different types of bacteria. The first one, nitrate, or NO3 minus. Okay. The second one, sulfate, SO4 2 negative. And the final one, carbonate, CO3 2 negative. So nitrate, sulfate, and carbonate. These are in order of their electronegativities. Nitrate is the most electronegative, carbonate is the least. This also puts them in order from best final electron acceptor, and what I mean by best is it gives us the most amount of ATP per molecule of glucose from those high energy electrons. Carbonate gives us the least, because of these three, it's the least electronegative. So microorganisms, some bacteria can perform both forms of respiration, depending on the environment that they're in. E. coli is a great example. E. coli we can get to grow very easily on an agar plate. That's being exposed to air, oxygen being one of the key components of that air. Bacteria will perform aerobic respiration when grown on an agar plate. Bacteria also grow in your gut. E. coli grows really, really well in the large intestine. Is there oxygen in the large intestine? There might be some, but it's minimal. And the reason why is that if you think about how we breathe or anything like that, we can get oxygen into our stomach, but most of that oxygen, since it's a preferred final electron acceptor, that gets used up quicker than any other of these final electron acceptors. So by the time we get to the large intestine, it's largely anaerobic. E. coli switches to a different form of respiration. They use nitrate as a final electron acceptor in this case, allowing them to still generate ATP. Just not as much as they would generate by aerobic respiration. Annie, go ahead.
Is fermentation?
We'll talk about fermentation on Monday, but there is a difference. Fermentation involves, I guess I'll just kind of precursor it. Fermentation involves glycolysis and nothing else. That's where I think glycolysis is really a decision point. At the end, and I'll back up to the slides here just to kind of show. All right. So at the end of this process, we've got a couple of pyruvate. This is a decision point for the cell. And in part, this decision is, quote unquote, decision is based on the environmental conditions. In part, it's based on what enzymes does the bacteria have. If they can perform fermentation, what we'll get is probably lactic acid, maybe ethanol, maybe other gases, CO2, hydrogen gas, other types of acids produced as well. If the organism can perform aerobic respiration or anaerobic respiration, the first thing is just to keep in mind, this shows you where all, oh, that does not show you, sorry. Here it is. Shows you where all the ATP is being made, where the NADH is being produced. Again, the numbers aren't important. What's more important is thinking about these in terms of themes. What is the purpose, or what is the goal of each of these processes? So here's what I want to do. I didn't plan to do this originally, but I think this is a good way to kind of cap up today. Glycolysis, Krebs cycle, electron transport chain / oxidative phosphorylation. Based on what's being produced, how would you define each of these? Or what is the major theme of each of them, I suppose is a better way to ask this. Come up with a word or a phrase that defines what the theme of each of these processes are. Takeaway point from each of these processes. Let's save glycolysis for last, but let's start with Krebs. What's the major theme in Krebs? We already answered that one today, didn't we? I was trying to see who would pay attention. Redox reaction is the major one. 2 for 2 on that one. What about oxidative phosphorylation? What's the theme? ATP? Any others? Oxygen, you could just say final electron acceptor. What was that, Emma?
Just the movement of electrons.
Movement of electrons. I would even, and I probably would just say like more redox reactions, right? So there's a lot going on in that one. Ultimately, what we yield from it is lots of ATP. Okay? And remember, the amount of ATP there correlates with these final electron acceptors. Oxygen will give us the most. Nitrate a little bit less, sulfate a little bit less, carbonate even less still. So the number that you've heard, oxidative phosphorylation, respiration produces 36 ATP or 38 depending on who you talk to. That's true when oxygen is the final electron acceptor. It's less for nitrate, but it's more than 2. It's less for sulfate, but it's more than 2. Less for carbonate, but still more than 2. Okay, so these kind of fall somewhere in between the amount that we get from fermentation or aerobic respiration. How would we define glycolysis? Or what would we say are the major themes of glycolysis? Breakdown of glucose would be perfectly fine. Production of pyruvate. If I were summarizing it, I think both of those work really well. I think the other term that I would throw in there, and I'm not sure how to definitely define this, but probably production of high energy molecules, or molecules, rearranging these molecules in ways so that you've got really high energy electrons. Because that allows for, pyruvate's an incredibly strong oxidizing agent, or I'm sorry, reducing agent. It's got high energy electrons that it's going to give to NADH and FADH2 in the process of citric acid cycle, TCA, Krebs, yada yada. Okay. So a couple of things just to finish up for today. We're pretty much back on track. Okay, so that's one. The one thing I would have liked to have done today, but we're going to push off until Wednesday. But I want you to take a look at this over the weekend, or one of these. There are a number of review questions that I've got that kind of review, one, the general process of the electron transport chain, but more importantly, this is a target to control microbial growth. So for example, if you have ever had an alcoholic beverage made with hops, beer, okay, or if you've just eaten straight hops.
I don't drink.
I didn't think so. I knew that from last year, I think, right? Yeah, okay. If you have hops have what we call isohumulones. They end up taking the hydrogen ions and allowing them to just pass right through the membrane without going through the ATP synthase. What the heck is that going to do to the production of ATP. There are other examples. Rotenone, really, really good for killing fungi. Block this step right here. Enjoy more of a human poisoning, cyanide? Great. Blocks this step right here. What effect do these have on the electron transport chain, and what effect do they have on ATP yield? Okay? So take some time to think about that. Try one or two of those practice problems over the weekend. We'll spend some time talking about it next week. I have quizzes from yesterday as well, so I'll try to get these back to you before you leave. Otherwise, I'll get them to you on Monday. Have a great weekend. Stay safe. Stay warm.
I feel like I did really